Today we'll be talking about the mechanisms of natural selection. Basically, how does an organism go through this process, what traits are selected as being the most fit, and what forces are going to drive the natural selection in a particular way. So we first need to analyze what populations are and have some basic information about populations. So a population is a group of organisms. It could be a group of warthogs, as you see in the one picture here, or a group of beetles, as you see in the other one. A gene pool is a combination of all the genes in a population. Actually, specifically, it is their alleles, the different versions of those genes. Remember that genes come in different varieties. So if we look over here at these beetles, what we have is many different varieties, actually 3 different varieties. We have the darkest ones, the lightest ones, and these medium colored ones. And so if we assign capital B to each of these dark brown ones, and lowercase b to each of these light colored ones, and capital B lowercase b to each of these medium ones, we would say that that is an example of incomplete dominance, and that would actually be pretty easy to figure out what the allelic frequency is. The gene pool would be the total number of genes in a population. So we have 1, 2, 3, 4 times 1, 2, 3, 4, 5. That's 20 total genes, or 20 total organisms, each of them having 2 genes, so that's 40 total genes in that population. And we could pretty easily look at this and calculate what the number of capital B and lowercase b genes are. Basically, each of these would have 2 capital B's, and each of these ones would have 1 capital B because they're capital B lowercase b. Oh, I'm sorry, these ones, this one, this one. These are the ones that would be capital B lowercase b. These ones would be lowercase b, so each of those we get 2 lowercase b's, and each of these ones would have 1 lowercase b. And so you could total those up and divide by the whole and get the allelic frequency. It's a percentage. It's actually expressed much like batting average. Batting average, if a person's batting .350, then that means that 35% of the time they're hitting the ball. That's actually pretty good. And so what we would look at here is the total number of capital B's and divide by the total number of genes, and that would give us a decimal number, and that would be the allelic frequency. For example, we look over here at these little pig things, and we have capital B, and this little pig here is capital B, lowercase b, and this one's lowercase b. The problem with this right here is this is a dominant and recessive trait. Remember, you can look at a recessive trait and it sticks out, and you can notice that very easily. But a dominant trait's a little bit more tricky because they can mask a recessive trait. It's difficult to tell a capital B and a capital B lowercase b just by looking at the pig. So let's say this was brown coat versus tan coat. So you either have brown or tan. So this one would be brown, 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 this one would be brown. And these other ones would be lowercase b. They're going to be tan-coated pigs. And you could easily calculate the number of lowercase b's that you can see, but some of them are going to be hidden. And so this is going to take a little bit more complex math to calculate out. This is where the Hardy-Weinberg equation comes in, and this is this population distribution right here, and right here. So this first line is going to represent the number of homozygous dominant individuals. This is homozygous recessive, and this one right here is going to be heterozygous. Let's talk real briefly about genetic drift, and we'll come back to the Hardy-Weinberg equation. Alright, so genetic drift, what that says is, at this time you have these pigs, they're actually known as peccaries, and so these peccaries are in an area, they're introduced, and before the peccaries are introduced, you see a population distribution like this, this is the number of cacti with the number of quills. And so you'll see down here that an average cactus has only 90 quills per plant. You know, some of them have 80, some of them will have 100. Very few have 130, 120. These peccaries get introduced to the area, and the cactuses that they're going to eat are going to be the ones with the least number of quills. And since these cactuses with 90 or fewer quills are being eaten, because they're the ones that are easier to eat, they're not passing on their genes. And so the ones that do have 110 and 120 and 130 quills, even though there's very few of them, those are the ones that are surviving and passing on their genes. So after a few generations, they take another survey of the area, and they notice that very few of the cactuses have quills in the 80 to 90 range, even 100 range, but most of them are up closer to 100 and 120 quills, meaning that the population has drifted in a particular direction. Think of it this way. Rabbits in an area such as we live in the summer, a white rabbit would stick out. Seeing a white rabbit would stick out, and it would be very, very easy to find that, and so predators would find it pretty easily. However, if there were a change in the climate conditions to where it was snowy most of the year, white rabbits would have an advantage and would not stick out as often, and they would be the ones that would survive more frequently and pass on their genes, and so you'd see a higher percentage of white alleles popping up in the population because they would have a better chance of passing those genes on to their offspring. Genetic equilibrium is when the allelic frequencies in a population stay the same. So basically, they've come to a balance. They will maybe go up a little bit, go down a little bit, but those allelic frequencies eventually balance out, even if there's something such as the introduction of the peccary to throw them off. Eventually, they reach some type of balance or equilibrium. The Hardy-Weinberg principle states that at genetic equilibrium, the alleles can be predicted. So this means that you can't have a major change in a population. But once a population has been stabilized for a couple of generations, what you can do is you can actually predict. And if you look over here, we have our typical Punnett square. And so that if you plug in this equation, p squared plus 2p cubed plus q squared equals 1. I realize that the 2's here are not represented as squares, but that is indeed what it is. P squared plus 2p cubed plus q squared equals 1. And so what this is saying is this p squared represents the number of homozygous dominant individuals, capital A, whereas q squared equals the number of recessive individuals, lowercase a, and 2pq would be the number that are heterozygous. Now, it's very difficult to predict this number and this number because this involves a dominant trait. Looking at these two different moths here, it would be very difficult to determine which one is homozygous or heterozygous without doing a genetic test. Even if you were to count all the moths in the population, it would be nearly impossible to say how many are homozygous, how many are heterozygous. But these moths are very easy to figure out because, well, they're white, they're recessive. It's actually, I believe this letter lowercase a represents albino. And so you count the total number of the whole population. So if you look at the whole population, 45% of the moths are dominant, and we'll get to how we calculate that later. 20% are heterozygous, and 35% of the moths are homozygous recessive. That equals q squared. And this distribution is then shown on this graph here, so that as the number of lowercase a goes up, the number of capital A goes down, and, well, there's this little curved thing here that represents the heterozygous. Okay, so if you look at this right here, this 0.35, you could plug this into the q squared right there and solve for q. Now, if you look at the end results, this is showing you the full equation and how we get to the end results. 55% of the genes in a population are the dominant gene, and 45% are the recessive gene. And so you know what P plus Q equals, it equals 1. It always equals 1 because that represents all the genes in a population. So P plus Q always equals 1. And so what you do here is if you plug 0.35 into Q squared and take the square root of it, it gives you a Q value of 0.45. Well, 0.35 plus 0.10 equals 0.45. And so since we know that Q is equal to 0.45, we can plug that into here, and we can also plug it into there, so we're left with P squared plus 2P times 0.45, because Q is 0.45, plus Q squared, that's 0.45 squared, which 0.45 squared equals 0.35 equals 1. So now we're left with P squared plus 2P times 0.45 plus 0.35. And then you could solve for P. We'll go over this in detail as we will be doing a couple of labs in the next few days on this concept. I do realize that at this point it is a little bit more difficult to grasp. Go ahead and review the crash course video where he discusses the Hardy-Weinberg principle. And like I said, we will be reviewing this quite a bit in class to try to get everyone to have the hang of it. Okay, populations and extreme genetic drifts. The founder effect is a sample of a population settles a new location. So if you look at here in a general population, 1 out of 20 individuals is even a carrier for a trait. In this case, if you look down here, this person has an extra digit there. They have an extra thumb. And so that's a pretty rare trait, 1 out of 20 people. Remember, 2 people who are carriers for a trait would actually have to meet up. And if you look here, there's no one in this population who even has that trait. So if this individual were to have children with any other individual, they're not going to have a child that actually has the trait. But what will happen a lot of times is they will find a new place to go to. For example, the Amish came from a relatively small population from Europe and came to Lancaster, PA, and even that small group left there and then traveled, and was a pretty small group, and traveled to the further west out towards Ohio and such, towards Holmes County, and then back towards Middlefield. And each time there's a smaller group that's moving on. But let's just take the original group. Let's say our original group, only one of those individuals out of 4 had that trait. Well, you've actually increased the number per individual. Up here it's 1 out of 20. Down here it's 1 out of 4. Now, let's face it, not a lot of people are signing up to join an Amish Ordnung. However, it does happen, but it's not the common thing to have happen. Over a couple of generations, that's going to stay as a 1 to 4 ratio. And as you do that, now you have more people who could mate and then eventually produce a person with polydactylism, having the extra thumb. And it's something that we notice. This is why you do see more genetic defects in some types of populations, like, for example, Amish. You don't have new genes flowing in, it's just these defective genes, because of the founder effect, show up more frequently. Bottlenecking is sort of a similar type of thing as the founder effect. In a bottleneck, the population shrinks, and so what happens here is imagine having a bottle full of these beads, and as the beads fall out, only a small sample of the diversity pops through, and so when you go back to a larger group, basically that genetic diversity has been cut down. This thing has happened with cheetahs. At some point in time during one of the ice ages, the number of cheetahs, there was a wide genetic diversity, but because of a bottlenecking effect where populations got really, really small, there's very little genetic diversity. And because of that, they're going to have problems recuperating. For example, their populations have gone really low, and this is of great concern because cheetahs already have reproductive issues due to bottlenecking. If you think of like rhinos, their populations have gotten down really, really low. Well, when they recover, they're not going to be anywhere near the diversity they were before. Some of those genes are just lost altogether because those animals are now extinct, more or less. I mean, not the whole species, but those genes are extinct because the animals that carried them are all dead. Humans actually went through a bottlenecking at some point in time. If you compare human populations to other populations of animals, our genetic diversity is actually relatively low. And this is a map showing the cheetah range around the year 1900 and current ranges, so you can see that because of bottlenecking and the reproductive issues caused by the bottlenecking in the ice ages, now their populations are going to have a real difficult time recuperating due to a second set of bottlenecking caused by humans. Speciation we talked about recently, and now we're going to talk about adaptive radiation. Remember, speciation is when you take one organism and it becomes two different species because they become less alike over time. Adaptive radiation is a similar idea, but you start with one and it fills many different niches and has many different species. So one becomes many. That's the big difference there, is that it fills many different niches. You know, some of these birds were good at feeding on nuts. Some of them were good at feeding on nectar, but they all have one common ancestor. Here you can see the finches that Darwin actually originally looked at, and you can see how over here they have the different types of foods that they would eat, even though they have a common ancestor, which is a finch. These are all finches, but they filled a variety of niches to expose different foods to avoid competition with each other. And here's just a picture of what his finches actually look like today. These are photographs of the different species of finch. So there are some debates among scientists about whether natural selection can occur gradually, such as in gradualism, or as in punctuated equilibrium, where everything will stay pretty even for a while, but then rapidly change. So here we have gradualism, where these two species become slightly different slowly over time. Other scientists say, well, what will happen is something major will happen and cause these two animals to go in different directions, and we'll end up with two different species, and maybe something else major happens, and now we end up with three species, and these changes happen relatively rapidly. Which happens? Well, probably a little bit of each. We know that animals do gradually change over time and become more and more distinct, but there are times when we do see periods of rapid evolution where animals become different quite rapidly based on something new in the environment, such as MRSA or some of these other super germs that we've seen recently. They weren't here before, and now they are here based on misuse of antibiotics, and they can pop up very, very rapidly and develop. We discover new bacteria all the time that are the result of misused antibiotics. Here's another diagram. This is sort of over a debate over how we got giraffes and okapis. Okapis are these animals that are related to giraffes. So here's your okapi and here's a giraffe. And in gradualism, basically, these two would become more different over time. The giraffes are more adapted to plains, and they have these long necks so they can get to leaves, where okapis have stayed in the forest, and they have adaptations on their legs, the striping, to help them camouflage. And their necks aren't as long because a long neck in a forest would just get caught in things. Other scientists have said that, well, maybe this offshoot happened very rapidly, and some of these offshoots over here happened more rapidly. Which is correct? Well, there's a debate, and as I've said before, it probably could have been some combination of the two. What is the result of natural selection? Well, the end result are these three terms here. The first one is divergent evolution. Think of things becoming less and less alike. Two groups of organisms become less and less similar. And if you think of dolphins compared to humans, we're both mammals, but yet we have very, very, very different adaptations. On the other hand, you have the opposite of that, convergent evolution. If you look at this dolphin here, which is a mammal, this ichthyosaur, which is a reptile, it's extinct. And here we have a shark. They have torpedo body shaped. They have dorsal fins. They have pectoral fins. They have tail fins. Similar adaptations for a similar environment. Same thing with the bat, the eagle, and the pterosaur. And also you see that among these different groups of anteaters. None of these guys are related. This one here is a monotreme, meaning it's an egg layer. This one is just sort of in a category all by itself. The giant armadillo is now extinct, but it's more closely related to armadillos. And then we have the giant anteaters of South America. It belongs to Insectivora. It's a type of mammal that is a placental. So that's convergent evolution, where they become similar even though they're not related because they're filling into similar niches. And then extinction is an organism does not exist anymore. The opposite of that is extant. For example, there are three extant species of anteaters and one extinct species of anteater. Extinction is the result of natural selection because basically the organism is no longer the most fit for the environment. A lot of times it's because the environment changes. So here we see that birds are not very closely related to reptiles and not very closely related to dolphins, but all three of these have flippers of sorts. They have torpedo body shapes. They are streamlined. They live in similar habitats. I would throw in the shark on this as well because the shark's actually very close. The difference is sharks can breathe underwater. These three cannot. But because they became similar, that's convergent evolution. But birds being different from reptiles and being different from mammals is divergent evolution. Diverge means to go away from each other or divide. Converge means to come together. Here are some examples of convergent evolution. Here you have a mole, and here you have a marsupial mole, not even closely related, yet they have similar adaptations. Lesser anteater and a numbat, which is kind of like an anteater mouse, and a marsupial mouse. The marsupial mouse has some interesting things about it. Flying squirrel and a flying phalanger, both low gliding critters, but again a marsupial and a placental. And there was even a Tasmanian wolf, which is now extinct, and a wolf, again a marsupial and a placental, filling similar niches, doing similar things in the ecosystem, but not even closely related. Here's an example of divergent evolution. Cichlids are found in Lake Victoria in Africa, and in this lake they fill all kinds of niches. You have fish eaters and snail eaters and algae scrapers, and they're all considered cichlids because they have the second set of jaws, but they've adapted to different roles in the environment because they were the only fish that's in this lake. It's biologically it's kind of secluded in a way, and so these cichlids live in that lake, but they filled many different niches because they were the only critters there, and to avoid competition they found different roles. And on this page we will talk about the different types of natural selection. The first one is stabilizing selection. So when you look at a population, generally speaking, you have a small percentage that are small and a large percentage that are average and a very small percentage that are big. And so if you look, for example, at human growth patterns, you have a few people who are under, let's say, 5 foot. Most people are somewhere between 5 and 6 foot. And then if you get all the way down here, you have your people who are like over 7 foot. And most people fit into this range right here, but over here you have your NBA players, and over here you have your very short people. And so if I erase all of this now. Alright, in stabilizing selection, though, what we do is we sort of throw some of those norms out. And small spiders possibly aren't going to be able to find mates. They're not going to be strong enough to fend off others and get a mate. The big ones are going to stand out for the birds. And so there's very, very few really small spiders, a whole bunch of medium-sized spiders, and very, very few very large spiders. So instead of your typical distribution where about 20% are really big and 20% are really short and 60% are in the middle, it's more like 90% are right in the middle and 5% are really small and 5% are really big. When you look at average population distribution, you would expect a bird to have a small beak, very few of them to have a small beak. Most of them would have average sized beaks, and very few would have very long beaks. But as a woodpecker, you use that beak to get to food. The more food you get, the better your chance of survival. And so what happens here is that the smaller beaked ones have less of a chance of surviving and passing in their genes, even medium-sized ones. Only the ones with really long beaks can survive and pass on their genes. And so the medium-sized ones die off, the small ones die off, and everything gets pushed in one direction. Now, it doesn't necessarily have to be big beaks. It could be small beaks. For example, it could be a smaller beak enables the bird to fly faster, and that enables it to pick up prey much easier. That would be directional in the favor of small. So it isn't always just big. It's one direction or the other. It doesn't have to be necessarily bigger is better or faster is better. All right, disruptive selection. So again, normally you would expect to find a few light-colored birds, a few dark-colored birds, and almost all of them being medium-colored birds. But that isn't what happens here. You have one island with very, very light rock, and you have one that's very, very dark rock. And so instead of having mostly medium-colored birds, what happens is the birds that live on the island that is very light in color, they survive there. The gray birds stick out in both places, so they get eaten up every time. And the ones that are very dark in color blend in on the volcanic island. And so you have light-colored birds on like an island that's mostly like limestone and such. They survive. Dark birds survive on a dark island. And the medium-colored birds get eaten both places. They don't pass in their genes. And eventually you end up with two populations, one that's light and one that's darker. And this is disruptive. It's going in opposite directions. You end up selecting for the extremes. The middle is lost. And actually, this could eventually lead to what we call divergent evolution. Now you end up with two different species because of reproductive isolation. There's nothing that's preventing the birds from this island going over to this island over here, going from here to here and breeding, but other than their chance of survival. One last thing to look at is what is known as sexual selection. And this is when Darwin just couldn't explain this. If you think about a peacock, that is a pretty useless type of adaptation to have a very, very large plumage. I mean, it doesn't make any sense as far as, passing on traits. I mean, what advantage is there to have a really, really, really big tail? It's just going to get caught and get in the way of things. But what Darwin had determined was, well, this gave it a breeding advantage because the ladies noticed them. And so that's sexual selection. Antlers. Yeah, they're used for fighting off predators, but they're also used for fighting off the deer from each other and battling and fighting for rights to mate. Some birds have very bright plumage that just makes them stick out. Again, easier for a predator to spot. But this also makes it so that they are easier to spot by the mates as well, and therefore have a better chance of reproducing. Okay, so coevolution is something that Darwin also looked at. And what we have here are two species that have evolved together in a symbiotic relationship. In other words, they have become very dependent on each other. They become so specialized that the evolution of one affects evolution of the other. In other words, we have a flower that's red. So the reason it's red is to attract hummingbirds. It's got this long tube, which makes it so that the hummingbird has to stick its long snout in there to get to the nectar. The hummingbird has developed a long snout to get to the nectar. It's the only thing that can get there. And then because of that, it gets pollen on the bird's face, and it helps fertilize other flowers. And so these animals organisms are dependent on each other. The hummingbird's dependent on the flower for nectar, and the flower is dependent on the hummingbird for reproductive purposes. And so they've become so tied together that if one becomes extinct, they will both become extinct.